Okay, I'm going to do a short video lecture on a little bit of review of topic number 11, which was DNA and chromosomes, and what chromatin is, chromatin packaging, and the structure of nucleosomes, and how basically chromatin is regulated. I got a lot of questions about this topic, and there was some confusion, so I hope this video lecture clears it up. So, one thing to remember to begin with is that inside the nucleus, you have a complex of DNA and you have a complex of protein. Okay, DNA is N complex with protein. Okay, and this is what we call chromatin. So chromatin is the complex of both proteins and DNA inside the nucleus. So let's take a thought example. If you take a nucleus here, right, and let's say you have a bunch of DNA in here, and this is your interphase DNA. All right, so this is when a cell is kind of expressing its genes and not really in dividing yet. And if you lyse the nucleus, so we break open the nucleus, what you'll find is a thick fiber about 30 nanometers in thickness, 30 nanometers of DNA and protein, okay, and this is chromatin. Sorry for my handwriting. And that's what this electron micrograph is showing you, is this 30 nanometer thick fiber of chromatin. Well, if you take each side of the chromatin here and you pull it, all right, so let's say you pull it as far as you can, what happens is you unravel the chromatin to show what we call beads on a string structures, okay? And what these are little beads of protein and DNA that are joined together by short pieces of linker DNA, okay? So DNA that's not associated with protein. And that's what this micrograph over here is showing you. So typically the interphase chromosome is in this kind of 30 nanometer thick fiber, but ultimately what makes up the chromatin is the smallest unit of structure, these beads on the string, okay? And these beads on the string are called nucleosomes. So nucleosome. Okay, and this is kind of the basic chromatin structure that makes up of all the chromatin in the cell. All right, so in summary of this slide, what I want you to understand is that DNA exists in a complex with protein, and we call that complex of DNA and protein, we call it chromatin. And the basic kind of building block of chromatin is the nucleosome, which we're going to look at the structure of next. So here's a nucleosome, and here's a nice picture of it up here. Okay, and a nucleosome is, again, a complex of DNA and protein. And the protein that makes up the nucleosome is called histone protein. Okay, a nucleosome is a complex of proteins called histones. Okay, and histones are interesting because they're a protein with a very high number of amino acids that are lysines and arginines. And if you'll remember, those two amino acids are positively charged. So a histone protein, let's just show a kind of a globular histone protein, has a very high proportion of positive charges, which means that the protein overall is positively charged. Well, if we remember, DNA is made up of the linkage of nucleotides within phosphodiester bonds, and therefore, because of its structure, it has a phosphate backbone that is highly negatively charged, right? So DNA is an overall negatively charged molecule, and the histone proteins are overall a positively charged protein. And so what happens is these electrostatic interactions is what allows the DNA to wrap around the histone protein. So if we have a bunch of histone proteins there, and let me just change this maybe to green, you can see the DNA with its negative charge is going to wrap around the histone protein, and that's going to be stabilized by the electrostatic interaction between the positively charged lysine and arginine amino acids and the negatively charged DNA. So a nucleosome, again, is a complex of negatively charged DNA wrapped around positively charged histone proteins, okay, and that's what kind of forms the basic unit of the nucleosome. So let's look more closely at what a nucleosome is. A nucleosome is actually a complex of 8 histone proteins, all right, H2A, H2B, H3, and H4. And you don't need to know exactly which histones they are. We're going to focus on H3 in a little while. But what I want you to understand is that the nucleosome is made up of what's called a histone octamer. Okay, and remember the octamer just means that there are 8 histone proteins. So you can see them down here. They're basically made up of 4 pairs of histone proteins that total 8 total proteins that make up the histone octamer. Wrapped around the histone octamer is 147 nucleotides of DNA. And this is about the length of 2 wraps around the nucleosome core. All right, so 147 nucleotides of DNA wraps around 2 times, so we'll write 2 times, around the histone octamer. And then the last part of the nucleosome is a short linker DNA. So if you look up here at this structure, here's our nucleosome, and then here's our linker DNA. It's a very short piece of DNA that links nucleosomes together. All right? And so again, this is the fundamental structure of chromatin, the forming of chromatin, which is the complex of DNA wrapped around histone proteins. So we've talked about basic DNA. You have your double helix that has no proteins. That double helix is wrapped around twice a histone octamer, okay? And those nucleosomes are joined together by linker DNA. All right, and that forms the basic fundamental unit of chromatin. That chromatin is actually compacted a little bit further. Remember when we talked about lysing the nucleus, you found this 30 nanometer chromatin fiber. Well, that's what you ultimately find in interphase nuclei. You'll find a 30 nanometer fiber, and this is dependent upon another histone protein called histone H1. Now, please remember that histone H1 is not part of the histone octamer. Okay, so it doesn't make up this core, but what it does is it binds to the linker DNA here, okay, and allows the 2 nucleosomes to compact further upon each other. So let's draw kind of a nucleosome here, and then a linker DNA and a nucleosome. And what histone H1 does is it binds to these linker DNAs, okay, and it basically allows the histones to pack together more tightly. Okay, so you can take your nucleosomes and now you can pack them tighter into this 30 nanometer fiber. Okay, so the formation of 30 nanometer fiber is dependent upon histone H1, which binds to the linker DNA. This is kind of the basic level that you're going to be at in interphase. Now, when the cell gets ready to divide, what it will do is it will start to compact these even further, and it will basically condense the chromosomes as tightly as possible, all the way down to the mitotic chromosome here that you're familiar with seeing during cell division. Okay, but most of the time your chromatin is going to be in kind of a 30 nanometer fiber, or maybe a smaller, or maybe a little bit more compacted than that. But typically your interphase chromosomes don't exist in these very tightly packed mitotic chromosomes. So we've talked about all the different levels of basically folding and compacting the chromatin can do. Well, I mean, that DNA has to be read. We want to express the genes in that DNA. So how do we actually access the DNA that's tightly wound around these nucleosomes? And I'm going to talk to you about 2 ways that this happens. And the first is by what's called modification of the histone protein itself. Okay, and this is where I want you to focus on histone H3. Histone H3 is a part of the histone octamer. And what's interesting about histone H3 is that it has the N-terminus of the protein is sticking out of the nucleosome. Okay, so if you imagine the nucleosome here, you'll have kind of histone H3 sticking out of it. And what happens to histone H3 is that there are lots of enzymes that come by, and they will covalently modify the amino acid side chains in this tail. So you'll have a tail here, right, and you'll have your nucleosome, and the tail's sticking out, and what will happen is a kinase might come by here and phosphorylate an amino acid on that tail. Okay, so histone H3 is subject to covalent modifications, and this will actually regulate and affects how tightly the chromatin is wound together. So let's look down here. Here's histone H3, and this is that N-terminal tail that we were talking about that's sticking out of the nucleosome. And each one of these amino acids, here's an arginine, here's a lysine, a serine, and this is showing you what we know happens to each amino acid. So this arginine gets methylated. That means a methyl group gets put onto it. This serine here is phosphorylated. Okay, this lysine here gets not only a methyl group, but can also have an acetyl group added to it. And so if you look down here, each one of these covalent modifications affects the chromatin structure differently. So for example, methylation of lysine 9 will actually turn genes off. It'll result in gene silencing because it actually will wind the chromosome even tighter so that the DNA can't be accessed. However, in this example, methylation of lysine 4 and acetylation of lysine 9 will actually result in gene expression. Okay, you need to know these specific examples, but you need to understand that covalent modification of this histone H3 tail can either turn genes off or turn genes on, and it does so by regulating how tightly folded the chromatin is. Okay, so a couple things to remember about this. First is that these covalent modifications are reversible. So not only can it be phosphorylated, but it can also be dephosphorylated. So you can turn a gene on and turn a gene off depending on what you want to do. These are all catalyzed by what are called histone modifying enzymes. So these are special enzymes that will do this. And like we talked about, the main functional groups that are used are acetyl groups, phosphate groups, and methyl groups. And they are covalently added to amino acid side chains. And this ultimately will control chromatin organization and regulates gene expression. So you can turn a gene on, you can make a protein by doing something to histone H3, and then if you reverse it, you can actually turn that gene off and stop making that protein. Another way of regulating chromatin structure is through a complex of proteins called chromatin remodeling complexes. Okay, and this is a complex of proteins that utilize ATP hydrolysis to physically unwind the DNA from the nucleosome. So you kind of think of it as your own hand. Think of your hand just grabbing this nucleosome and you're turning it so that the DNA will unwind itself. And that's what this protein is doing, or this complex of protein is doing. And it's using the energy of ATP hydrolysis to unwind the chromatin and therefore to allow the genes to be accessed and possibly expressed. Obviously, during mitosis, when the cells get ready to divide the chromosomes, the chromosomes are in their most highly compacted form. That means that all of the chromatin remodeling complexes are typically inactivated so that you don't unravel what you've already raveled back up in order for the chromosomes to divide. So 2 ways I talked to you about histone H3 covalent modifications and chromatin remodeling complexes. These are 2 ways that cells regulate chromatin organization and will ultimately can regulate gene expression. So there are 2 main forms of chromatin in cells. You have heterochromatin and euchromatin, and we'll talk about each one. Heterochromatin is the most highly condensed form of chromatin in the cells. Okay, it's kind of the most tightly packed the chromatin can be. And typically you'll find heterochromatin at the ends of chromosomes here where the telomeres are, and you also find heterochromatin where the centromere is in the chromosome. Okay, so heterochromatin is regions of the chromosome that either have no genes inside of them or actually are turned off. They're regions of the DNA that just cannot be accessed because they're so tightly packed. One thing I do want you to remember about heterochromatin is that classically methylation of lysine 9 has been associated with heterochromatin formation. So if you methylate lysine 9 on histone H3, you'll basically turn all of the chromosome or all of that region of the DNA that's wrapped around that nucleosome into heterochromatin. Okay, so one covalent modification can basically turn DNA into heterochromatin, and any gene within that region is automatically turned off. And euchromatin is basically the opposite. Euchromatin is essentially any DNA that is not heterochromatin. It's typically looser, it's less compacted, it's accessible by proteins that need to transcribe genes, and so therefore in euchromatin you'll find gene expression. Okay. So if I ask you where would you typically find a region of the in which part of a chromosome would you find genes actively being expressed, that would be euchromatin and not heterochromatin. I gave you 2 examples of how chromatin structure regulates gene expression. And the first example is a phenomenon that's called X inactivation. Okay? And this is essentially gene silencing of X chromosomes in females. So if you have a fertilized embryo here, okay, you get basically an X from your dad and an X from your mom. And what happens is, as that cell divides, randomly one of the X's is turned off completely. It's silenced for good, okay, because you only want to express the genes from one of the X's. You don't need both of them. And it's a random process, and so you can see here each cell has turned off one of the X's permanently. And as that cell divides, that same X chromosome will be permanently turned off for the life of that cell and all of its descendants. Okay, so every cell in your body has one of these X's inactivated permanently. Okay, and the way your cell does this is by regulating the chromatin structure. It goes through a signaling process where it tells this X chromosome to fold completely into heterochromatin. Okay, it packs it up as tight as it possibly can, and therefore the entire chromosome is inactivated. So X inactivation in embryos and in early cells happens through the formation of heterochromatin of one of the X chromosomes. And it's a random process, and it's not really known exactly how cells decide which X chromosome to inactivate. The other example I gave you was a mutation example of what would happen if you had a gene that was in euchromatin, so therefore it was actively being expressed, and all of a sudden there was some kind of structural rearrangement in the chromosome where that gene was put into a region of the DNA that was supposed to be heterochromatin. Okay, so to show that, I'm just going to draw very briefly. Let's say you have a heterochromatin region, euchromatin region, and then a heterochromatin region again, okay, of a cell. And this is gene A, all right, which is making a protein. Well, what happens if all of a sudden a piece of gene A, through some kind of structural mutation or rearrangement in the chromosome, which does happen, gets put into the heterochromatin region? And so now this whole region is turned off, and so what you'll find is that you'll have your heterochromatin, you'll have your gene A, you'll have your missing piece, okay, so you basically still have your euchromatin there region, but now you have gene A, and then in here you have a fragment of gene A, okay, fragment of gene A. And so what's happened is you've made a mutation where you've deleted part of gene A here that's being expressed, and you've essentially moved it somewhere else that can't be expressed. And so now this gene could be possibly mutated or not functional, or for example, the entire gene can be moved into a region of heterochromatin, and therefore that gene is permanently turned off. Okay, so every time your cells divide, these kind of structural DNA rearrangements can happen, and if typically a euchromatin gene is moved into a region of the chromosome that is supposed to be heterochromatin, that gene effectively will be silenced, just like the X inactivation example. Okay, so the regulation between euchromatin and heterochromatin is very crucial because it ultimately will regulate which parts of your DNA is being expressed and what regions of your DNA are not being expressed. One last thing we did in this lecture is talk about the inheritance of chromatin structure. So for example, let's say you have a region of DNA that's highly compacted and it's in its heterochromatin form. Well, as that DNA divides and duplicates and the nucleosomes reform around that DNA, you're going to have some nucleosomes that had the covalent modification on them, and you're going to have some nucleosomes that formed fresh because you needed to make double the amount of nucleosomes because you've doubled the amount of DNA. And these nucleosomes don't have the covalent modification. Well, somehow your cell knows that there should be covalent modifications on these nucleosomes, and after your DNA has been replicated and folds into its chromatin structure, enzymes will come by and will covalently modify all of the nucleosomes that were modified prior in your parental DNA. Okay, so simply what this says is that covalent modification of histones, which ultimately affects chromatin structure, is copyable, is inheritable. Your cell knows that this nucleosome was modified, so therefore when your DNA divides, it'll copy over that information, and therefore your chromatin structure will be the same in each daughter cell. So again, that's why when the X chromosome is inactivated in the early embryo, every descendant cell from that early embryo, the same X chromosome is inactivated because the cell knows that that chromosome is supposed to be in a heterochromatin state. So just remember that these things can be inherited, from the parental cells as your cell divides. And in fact, we talked about that there's a new field of biology now out there called epigenetics, and what they're finding is that a lot of gene regulation is occurring on the level of what's called the epigenome or the epigenetic level, which means on top of the DNA. So what they're finding is a lot of gene expression is being regulated by covalent modification of the nucleosomes, and that this is inheritable, and that they're finding that these inheritable changes of the nucleosome are affecting gene structure and ultimately gene expression. And so here you'll see a key term called the epigenome, and that scientists are currently putting together all of the known histone modifications within chromosomes of an individual, and therefore not only do you have your genome, which is the combination of all of your chromosomes, but you have your epigenome, which is the combination of all of the modifications of those chromosomes as well. So I know that this topic can be somewhat confusing and complicated. I hope this cleared it up. Please again send me emails or see me in class if you have any other confusion about these type of topics. Okay, but the key to this lecture is to understand the basic structural unit of chromatin, which is the nucleosome, know its structure, know how it's modified, know what the effects of those modifications are, and know why it's important to have parts of your chromosome as heterochromatin and parts of your chromosomes as euchromatin, because ultimately these are regulating the expression of genes within the chromosomes.